kappa chain successfully on one of those chromosomes. Okay, basically you have deployed. So if you are able to rearrange it on one of the allele, so basically this gives a signal so that rearrangement does not take place on the second allele, right? This is called allelic exclusion because you don't require that. And for example, at some time point, so if you have a problem in rearranging at one allele, then your cells give you a chance to rearrange um, for making this receptor by productively rearranging the second allele. Okay, both for heavy chain and for light chain. So essentially, what it means is, um, whenever you are done with a successful rearrangement, either for heavy chain or for light chain on one of the allele, so the other allele does not allow or does not basically rearrange that segment. This is what it means. Okay. Obviously, this occurs both for heavy chain and light chains, right? So, and the combination could be that you, in some cells, you are able to rather rearrange both heavy and light chain from maternal side. In some cases, you might do it both for paternal side, and in some, one maternal and one paternal side. Okay, so all these possibilities exist. And why this process happens is because you have to give your cells a chance to make every possible arrangement to assemble or to rearrange these VDJs on surface of the in the chromosome so that you can actually generate these receptors. This is what allelic exclusion means. So consider the situation here. Okay, so as you know, many situations can arise. So let's say if you have a productive heavy chain allele on number 1, so this gives a signal that you do not have this rearrangement occurring at allele number 2, right? And after this heavy chain, then you start with this question. Anyone has a question? No. Okay. So once you are done, because you have gotten a productive rearrangement on the heavy chain allele. Okay. Obviously, this has to start with the light chain rearrangement process afterwards. Okay. And then you, for example, try to rearrange this light chain gene segments, right? And if you are able to do it successfully at allele number 1, so you don't require this to be happening on allele number 2. And obviously, because you already have in one go, so you have arranged heavy chain and light chain on one of those alleles. So you don't actually require it to be rearranged on the second allele, both for heavy chain and for example, in this case, lambda chain on the allele number 2, and obviously you don't require kappa chain to be rearranged now either on allele 1 or 2, because you already achieved that successful rearrangement for heavy and light chain, right? Scenario 2 could be that you have a non-productive heavy chain allele rearrangement on one of those alleles. Then you start with the second allele, right? And if you get a successful rearrangement there, then you start with, again, the same situation where you are rearranging these genetic segments on light chain. And for example, if one is successful, so you don't require these two. The third situation could be that you have a productive on allele 2, but non-productive light chain on allele number 1. Then obviously you start with allele number 2 for lambda chain. And if you get a successful outcome here, so you stop this process of rearrangement there, right? Now, how this stop signal is given and this loop, right? So remember, in that video it was said that these loops, they do not have any further role in the process. Actually, it was later on shown that these loops actually give a signal that you don't have to rearrange on the second allele. The fine detail of these processes are not known, but they actually are somehow involved in giving this stop signal so that rearrangement stop on the second allele. Okay. So now you can come up with all kind of possibilities that exist to arrange both on heavy chain and light chain for both lambda and kappa, right? So productive rearrangement means, so you have successfully rearranged these segments, VDJ for heavy chain and VJ for light chain, right? Both for heavy and light. So this is basically productive means, right? And if obviously both heavy chain alleles are non-productive, B cell has to die, because without rearranging heavy chain, so it cannot really proceed to the light chain rearrangement. Okay, so it cannot really proceed to the next step because this is happening one after the other. So obviously, if heavy chain fail to rearrange on any of these two loci, so the cell has to commit suicide, right? And if heavy chain rearrangement is productive, but then light chain is non-productive, again, same outcome, cell has to die. Okay. So now you kind of know that you exhaust all these options in a sequential manner to make it possible that your cells are able to rearrange a productive process wherein you can actually have a rearranged DNA for heavy and light chain, and eventually, so these things are transcribed and translated and exported onto the surface, serve as BCR for B cells, and obviously beta and alpha chain for your T cells, which will generate a productive TCR. Okay, this is not to say that this still has to this will still survive. Okay, because then it will undergo this process of selection, where if it is autoreactive, the cell is going to die. If it is not autoreactive, this is going to select out. Okay. Okay, so this is one situation. Rather, it kind of talk about all these possibilities. So you start with progenitor cells, which start with the D and J recombination. In this case, so for example, everything is non-productive, right? So let's say if you have this VDJ recombination happening, and on allele 1, this is non-productive, and it starts with allele 2, and if this is again non-productive, the cell has to die. Other situation could be that you have a successful rearrangement only in one go, where you have first rearranged this heavy chain. Now the cells start expressing IgM or mu, right? And then in one go, so you are able to rearrange for the light chain, in this case kappa. And as you know, so this actually is a random process, so you don't have any specific sequence for kappa and lambda. Okay, so it can be either lambda or kappa through this VJ recombination, and you get a productive heavy and light chain rearranged, which will give rise to this membrane bound immunoglobulin, right? Obviously, because this mu chain is involved, so you call it as IgM. Okay. Other thing that can happen is obviously you have rearranged productively this mu. Okay, and then you start with let's say on allele number 1, this is non-productive, so then you give a chance to do it for allele number 2, and if now this is productive, so then it will start with one of those heavy chains, and if it is not successful, then it go to the second heavy chain, which is lambda, right? And if lambda is successful, good. Yes, you have to go through different steps. So this is not happening in one step, but nonetheless, you are able to rearrange both heavy and light chain. Okay, so all these processes basically make sure that you try to give a chance to each developing cell so that they can productively rearrange specific receptors when they were developing in bone marrow. Obviously, the same thing has to happen in thymus for T cells. Is that clear? So allelic exclusion, as the name suggests, you have to exclude the other allele, and you exclude only when you have achieved a successful rearrangement on one of these alleles. Then the second is excluded. This is what allelic exclusion means.
So,
Yeah.
in case the stop signal doesn't work in between and both the alleles are actually getting a chance to rearrange, then what happens?
Well, you can do it artificially. Okay. For example, if you have a mature B cells, but in developing cell, this is not going to happen. Okay. If you have mature B cells through lentivirus transduction processes, you can actually transduce a construct which has a rearranged other chain, and then you try to find out how many of these receptors are expressing. For example, if you do it for light chains, how many are expressing lambda and how many are expressing kappa, right? So all those are artificial situations. But what we were talking about here is a natural situation. Okay, naturally that does not happen. What you are saying is not a natural situation. Okay, natural situation is allelic exclusion is happening. Okay, artificially you can try to do it in your mature B cells or T cells by transducing both whatever alpha and but that is a chimeric situation, and how efficiently these cells are going to perform their function is questionable. That's one. Second is maybe these cells become cancerous and this actually has been shown in some studies that if you try to make a chimeric B cell which express both alpha and beta for T cells and for heavy and light chains, and if you try to rearrange all these non-natural combinations, cells are not physiologically relevant. Either they start dividing so rapidly that they end up becoming lymphomas, or somehow they die in this process.
Fine, sir.
Okay, so these are different ways how by which you can generate antibody diversity. Obviously one of these things is you have multiple germline gene segments, combinatorial diversity provided by VDJ recombination, junctional flexibility where you add some, delete some nucleotides, and if these are still able to generate in-frame product, so those basically will provide you with novel specificities. And then you have P-region nucleotide addition and N-region nucleotide addition. Once these cells actually have undergone these processes, obviously, so these cells can undergo this process that we call as somatic hypermutation. but somatic hypermutation occurs only in the variable region uh, and only in B cells. It doesn't occur in T cells. Okay. This also it will not change the specificity, but it will actually make it more or less affine. Okay, that's what somatic hypermutation means. And obviously, a combinatorial diversity provided by both heavy and light chain. Um, so, for example, you have one combination where the light chain actually or heavy chain is same, but light chain is different. So this will start binding to other antigen. Okay, so because of this process, so you have the capability to recognize, you means through your B cells or T cells, recognize all kind of antigens available, right? All right. So then there are subtle differences in some species. So with that, we are not going to talk about. But essentially, the processes remain same. Okay. For example, one example rather I gave you was for this unique heavy chain only antibodies. Then you would consider key these animals, all these camelids are also vertebrates, so they have heavy and light chain, obviously. But then how come only the heavy chain is coming onto the surface of these B cells? Okay, those are the questions that people are still trying to find an answer to. There are some data which suggest that this is a possible mechanism, but these are all those evolutionary changes that have happened. I don't know what will happen even in humans. I don't know, after maybe several, I don't know, whether we actually live for million years. Uh, but all these possibilities do exist, that because of this continuous evolution of these loci, uh, and as we know about evolution, so this always try to give a fitness, and if there is a fitness associated with an evolutionary change, why not go for it? But essentially, this is what we understand about the generation of antibody or the TCR diversity, right? This is one example, again coming from your book, QBs, where how actually you estimate, okay, what kind of different combinations you can generate. Combinatorial antibody diversity in human and mice. So this is shown for human and this is for mice. And what essentially you do is you find out how many V segments are there, how many D segments are there, how many J segments are there in heavy chain, and how many uh, V and J are present in light chain for both kappa and lambda, right? Obviously, you don't have any D in light chain, so it's D is actually 0, right? Now, how do you calculate that? How many different combinations can take place? You just multiply these numbers, and you get to whatever this 6,000 or whatever number, right? Same for one of those kappa chain, one of the light chains, and another for lambda, right? And then if you multiply this by either this or this, rather it's not either, it's basically and, because in some clones, so you arrange kappa, in some you arrange lambda productively, right? So you have to add up these two numbers because both these things are not happening. So this is not a multiplication here, this is addition. Okay, so you add this number with this number, and whatever you get in bracket, right? Let's say you get whatever this x, you multiply that x by whatever the number you are getting from this VD of heavy chain, and this is the diversity that you are able to generate for what? For BDA recombination in humans, right? And this is not even talking about those extra nucleotide added or removed, or p nucleotide or n nucleotide, pseudogenes, gene conversion. All those phenomena are not talking or are not considered here in this table. Okay, just by a simple math, so you can generate millions and millions of specificities, and obviously, as we showed in one of those slides, the estimated number uh, was close to 2 into 10 raised power 15. So that means the other processes are also actually playing a major role, right? Now, one obvious question that you can always ask is, for example, one species, if it has less number of these VD and J, right, or B and J for light chain, so this will have limited ability to recognize different antigens, right? Uh, and that actually can be accounted for by evolutionary advantage or disadvantage, right? So this is exactly the same thing done for mouse, right? So did you understand this table? So you multiply these VDJ, you multiply this one and this one, because in your antibody molecule, you can either have kappa or lambda. So you add up these two numbers and multiply that by whatever the number you are getting from heavy chain. And that is a functional diversity you can achieve just with the number of VDJ segments. Okay, obviously there are other mechanisms to make it more diverse, and these mechanisms are also playing a major role. And also it was shown, again, let's not complicate these things, but in recent times also people actually have shown that as you grow older, right, so somehow you tend to have uh, limited repertoire of these cells, because these cells keep on dying, and your primary lymphoid organs are not that functional anymore, right? So then these other processes kind of take care of, to some level, what diversity you are able to achieve. And one of the dominant mechanism actually is this TDT activity. Okay.
Excuse me, sir.
Yeah.
Sir, could you please explain once again why are we adding kappa and lambda light chains?
Yeah. So who is going to explain? Rahul. Rahul Sharma. I can see this name in the participants. Can you explain? Or you basically logged in and then you're not seen? Anjana? Can you explain why we are adding? Why we are adding this number and this number?
Sir, I get it that either the light chain will be present, either it will be kappa or lambda, but I'm not able to sort of comprehend why we are adding.
See, when you are saying that in one cell, it can be either kappa or lambda, okay? But if you talk about the other cell, again, the same combination, either kappa or lambda, right? So this diversity is calculated on the total number of B cells you are able to generate, right? And total number of B cells which express one specific receptor or the other specific receptor, because obviously, so you cannot really multiply these numbers, because either you use kappa or you use lambda. Okay.
Right, sir, got it, sir. Yes, sir.
But one cell, for example, has kappa, another cell might have lambda. So that is why this addition is happening. So this is not multiplication as such.
Yes,
So
sir.
for example, if you have a BCR, rather the number of BCRs, it's not like one BCR present on a particular cell. So there are thousands of those, right? And out of those, let's say you have almost 50-50 distribution or 80-20 distribution of kappa chain utilizing or lambda chain utilizing receptors, right? Then you can try to multiply it, but that is not what is happening, right? And one of the theory that was actually proposed by F.M. Burnett way back in 1950s is that every cell that you have, right? So you have one cell, you have other cell, you have other cell. Whatever the number of receptors, whatever the identity of that receptor you have on this cell, okay, so that will remain same for all the receptors on this cell, okay. For example, if you have let's say you come up with any combination, V1, D1, J1 for heavy chain, and V1 and J1 for light chain. So all the receptors will have exactly the same recombination. Okay, it cannot have a different recombination on one particular cell. Obviously, for other cells, so this might be different. So all the receptors, all several thousand receptors that are present will have exactly the same antigen specificity.
Right, sir.
Yeah. Okay, Rahul,
So,
did you understand what I said? So you wanted me to repeat?
Yes, sir, I did.
Who had this question? Vishnu?
so why is the number of B segment decreased in the mice from 134 to 101?
No, which one are you saying? 130, which one? 134 to?
So 101, when we are multiplying, it's 101.
I think this is wrong. Okay, or it could be that some of these are pseudogenes, which are actually pseudo segments, which are not utilized. Okay, but essentially, the number has to be a multiplication of these segments, which can maybe this is a mistake. Actually, this is a mistake, I think. If the only way I can think about it is that somehow some of these genes actually are gene segments are pseudogenes, and they are not really able to be involved in the process of recombination.
But sir, I think it's a mistake only because below they have clearly mentioned that only.
Well, I don't know. So, well, try to calculate. Let me try to put a quick maths here. So,
sir, it's same. I mean,
if you multiply 134, 13, and
no,
4.
sir. 101 multiplied by 13 and multiplied by 4 is 5252. So
yeah, then it could be 134 has to be 101.
Maybe so. Fine.
Okay. Well, as I said, so the other explanation could be that these pseudogenes somehow are not really involved in the process. And that is why, if you actually calculate the unique sequences, you might have 134, but 101 are actually being utilized. Okay, just find out more about it.
Sure, sir.
All right. Somatic hypermutation. So this is one of the processes that remains to be discussed. Basically, mutations occur much more at much more higher frequency in the genes than other genes. The genes means these are genes which are responsible for encoding your BCR or TCR. Okay, and obviously, the somatic hypermutation actually is occurring when antigen is seen by these cells. Okay, invariably, this process actually is taking place in secondary lymphoid organ, and the site, basically where these B cells see these antigens and they start expanding in numbers. So this site actually is called germinal center reaction. Obviously, at this site, so your B cells also get signals from T cells either through cytokines or through co-stimulation or through other molecules. Cell-to-cell interaction is happening at this site, right? And this process of cell-to-cell interaction, or these B cells sensing some of the cytokines, induce this process that we call a somatic hypermutation, right? Hypermutation means basically, for example, in your hypervariable region 1, 2, 3 or CDR 1, 2, 3. So those residues which actually interact with the antigen, somehow they tend to mutate in such a way that you make it more affine interaction. Okay, the interaction actually is made more affine. And those clones are the one which are selected in the process, and they expand more during the response. Okay, obviously in the primary phase of the response, so this is not happening so dramatically, but when you keep on doing this boosting, so this process actually is observed much more vigorously than you get it for the primary immune response. Obviously, all the cytokines are playing a major role in this whole process. So I'll show you again. I'll try to run a video so you understand how this process is happening. Class switching. Class switching actually is, we already have mentioned it, that you have the same variable region on the antibody molecule, or for example, rather, it's only for the antibody molecule because you don't have a class switching in your RT-PCR, right? Same variable region, right? Initially, it was, let's say, hooked with Ig mu, and once these cells were activated, they produce Ig M type of antibodies. Okay, but receiving these signals from the environment, and the cytokines are the major player in this process. So then this mu is replaced by gamma, depending on what cytokine you have, or this alpha or epsilon, right? So these chains of constant region of the heavy chain with the same specific region in the variable region is called class switching. And then you generate these different classes, IgM, IgG, IgA, IgE, all those, right? Obviously, this process, again, is happening after antigenic stimulation, right? We know that the preceding step actually is a productive VDJ recombination, right? And once you have this recombination, so then your constant region actually is attached, and then initially you make this Ig mu antibody, rather IgM antibody, Ig delta, and IgG, IgA, and IgE are because of this antigenic stimulation, right? The enzyme which is critically involved in this whole process actually is called activation-induced cytidine deaminase, AID. Okay, one of the cytokines that actually is pretty well defined to do this process is IL-4. So let's try to run this video and see if it makes more clear case. Okay, so let's try to just click on it.
The browser you're using is allowing your behavior to be exploited.
Okay.
These cells can only express surface isotopes. In these cells, the heavy chain has previously undergone VJ recombination that placed rearranged D and J segments upstream of the Cu and C delta constant regions. Transcription initiated upstream of the rearranged VJ segments can produce alternatively spliced mRNA transcripts that generate either IgM heavy chain or IgD heavy chain proteins. After translation, the functional IgM and IgD proteins associate with light chain proteins and become expressed on the cell surface. In this initial orientation, transcription may be compromises only for the upstream of the and produces only IgM and IgD isotopes. The chain segments encoding the constant region for all isotypes are located downstream of the Cu and C delta constant regions. In the mouse genome, these constant regions are C delta 1 2, C gamma 2A, C epsilon, and C alpha. With the exception of C delta, each of these constant regions is preceded upstream by a sequence of DNA known as a switch region. Switch regions range in size from a few kilobases to nearly 10 kilobases in length and are composed of hundreds of repeats of G-rich short DNA sequences characteristic of these regions. For example, the switch region located upstream of the C-mu constant region is approximately 3 kilobases in length and is composed of approximately 150 copies of this short 21 nucleotide code, which itself contains 3 copies of the pentameric sequence GAGCT. Located upstream of each switch region is a DNA sequence that functions as a promoter called an I region. Under appropriate circumstances, the I region promoter may be used to initiate transcription by RNA polymerase II. Transcription begins just upstream of the switch region and continues through the region of DNA including the constant region. When this first occurs, the messenger RNA produced is known as a sterile transcript, initiating the I region, and therefore cannot be translated into functional protein by the ribosomal machine. Isotype switching is stimulated in the course of an immune response. An activated T follicular helper cell engages a B cell through linked recognition. The follicular T cell expresses CD40 ligand and produces cytokines, in this example interferon gamma. CD40 ligand engages a CD40 molecule on B cells. These transcription factors cooperate to selectively induce the transcription of the I region promoter upstream of the C gamma 2A constant region. However, they do not induce transcription from other I region promoters, such as C epsilon or C alpha. The different transcription factors activated by the cytokines determine the specificity of I region activation. In this case, activation of STAT1 by interferon gamma leads to the activation of I region for C gamma 2A. Induction of transcription in the I region promoter opens up the DNA and allows an enzyme known as activation-induced cytidine deaminase, or AID, to access the switch regions. As RNA polymerase II transcribes the sterile transcript, the elements in the switch region induce a behavior in the polymerase that allows AID to deaminate single-stranded cytidine residues in the DNA and convert them randomly to uracils throughout the switch region. Through the action of 2 other enzymes, uracil DNA glycosylase, or UNG, and apyrimidinic endonuclease, or APE1, these uracils are eventually converted into single-stranded nicks in the DNA on both of its strands. These processes also occur at the CMU region or any other switch regions actively transcribed in the B cell. These activities create multiple breaks in the DNA of the mu switch region, as well as the DNA of the switch region selectively activated by cytokines. Because the breaks have been produced in 2 distinct locations at a great distance from one another, the double-strand break repair machinery joins the 2 switch regions and excises all of the DNA located between the 2 damaged regions. After the double-stranded DNA break repair machinery finishes its work, the damaged switch regions will have been spliced together at some random point, leaving remnants of each switch region on either side of the repair joint. The rearranged VDJ segment is now placed a few kilobases upstream of a new constant region, in this case C gamma 2A. This now makes it possible for transcription initiated from the V region promoter to generate a transcript encoding the original variable region, but now in the context of a different constant region. When the messenger RNA is fully spliced, the intervening switch region sequences are removed, leaving only the segments encoding a fully functional C gamma 2A heavy chain. The B cell that has undergone isotype switching will begin to secrete antibody of the new isotype class.
Okay. Is it clear? If not, so if I upload these slides again, you can basically put this in your URL and go through this process. There are other videos also which actually make it more comprehensible. But this gives you an idea basically how this class switching actually is taking place, where you have the role of cytokines, you also have the role of cell-to-cell interaction, right? B cells interact with a particular kind of T cells, particularly the T helper cells. Okay, and these interactions are responsible for making one isotype or the other isotype. See, in many situations, for example, at mucosal infection, so you require IgA, which actually, if produced by your B cells, can actually pass through this process from some mucosa to the mucosal sites, and then try to neutralize the pathogen or whatever the antigen that you have there. So the cytokine, which actually is responsible for making this happen, is TGF-beta, right? So now, based on these possibilities, you can actually try to rewire your immune system in a way that you can either produce IgG type of antibodies or IgA type of antibodies, right? So is that clear? So basically, what how this class switching is happening? I can understand that maybe some of you are not able to hear what was actually said in this, what was actually articulated in this video. What you do is, again, run this so that you can understand it more. Okay, so essentially, this is what it means. So you have already rearranged your VDJ. And if you have to, for example, remove this mu, and then you have to start producing, let's say, gamma 1, right? So you have the looping of this DNA, so this is removed, and then this VDJ actually come in contact with G. You start making IgG type of antibodies. Now, if you change the cytokine, what you are exposing yourselves with to Ig, let's say TGF-beta, so then these antibodies can actually, these cells, they can start making the same antigen specificity, but with Ig alpha. Okay, so you can actually make IgA antibody from this. The sequence remains. The first one is IgM, obviously IgD, which actually are part of your B cell receptor, then G, then E, then alpha, right? Obviously, you can actually fine tune these processes just by providing the signals required for class switching. Okay, so yeah, we also did talk about that there is actually a lot of similarity in the rearrangement process of BCR and TCR. And we know that T cells actually come in two different players, either they are alpha and beta or gamma and delta, right? Alpha and gamma, so they have VJ and then followed by the constant segment. Yes, obviously you don't have different class switching mechanism for TCR, so you have only one constant region, right? And for beta and delta chain, so you have VDJ and then constant region, right? Now you can know from this that beta and delta are similar to your heavy chain in BCR, right? And alpha and gamma actually are similar to the light chain in your B cells. This is what happens for a TCR. So you have, let's say if you talk about alpha and beta. So you have beta, the first thing has to start with this beta, one of the D combined with one of the J's. Once you have a D and J, so then one of the V's come and complete this process of VDJ recombination. This is combined then with a concentration, right? And you, after transcription and translation, assemble the beta chain of your T cell receptor. Same thing is happening for alpha chain, with the exception that you don't actually really have functional D segments there, right? Again, same thing for TCR, and this is for for immunoglobulin or the antibody molecule. VDJ for beta and delta for TCR. For heavy chain, this is VDJ. For alpha and gamma chain, so this is VJ, and for light chain, this is BJ, right? All these things are also true for your TCR, okay? Where actually you have two chains providing you the specificity of TCR. You have P nucleotide addition, you have N nucleotide addition, you have junction flexibility, VDJ recombination, multiple gene segments. But one process that you actually do not have in TCR is somatic mutation, somatic hypermutation. Okay, and probably the reason for this is that these cells actually are developing in thymus, and once they have passed through this process of selection, you really cannot afford to change the T cell receptor expressed by these cells, because then they can be potentially self-reactive. The same thing can happen for B cells, okay? But for T cell, for most autoimmune diseases, actually, this is predominantly driven by T cells, okay? This is one assumption that people might think, that TCR does not have the somatic hypermutation because you cannot really afford to make autoreactive T cells to avoid autoimmune diseases. Okay, the other topic that we have to talk about actually is antibody engineering. Okay, so that we will do tomorrow. Briefly, we'll talk about this topic. Okay, so it's a very big field as such. So, but basic steps, what are involved in antibody engineering, we are going to discuss tomorrow. All right. So let's end this thing here.